Hello and welcome to another video in the lecture series Advanced Biomaterials. So in today's video we'll be looking at a very interesting concept or a very interesting material which is known as smart polymer. So smart polymers, what are they? So smart polymers are such polymeric systems or such polymers which can show a observable or a detectable change in their properties which can arise because of change or because of application of a particular external stimuli or change in an external environment or an external condition. So a smart polymer is a polymer which shows change in their properties arising from an externally applied stimulus. Now, these change in properties can be change in their shape, their volume, their phase, and also changes in properties such as conductivity or in their or viscoelastic properties or mechanical properties. So, but we need to remember that these changes are brought about by externally applied stimulus. So these stimulus which will be applying to them, and in response to those stimuli these polymers will show some change in their properties. So externally applied stimulus can be temperature, pH changes, and even there are different polymers which show changes in properties when applied to an external electrical field. So electrical fields, optical properties, and even visible light. So these polymers can have different stimuli in which they will be response to which they will be undergoing changes in their physical properties. So an example of such a polymer or such a smart polymer is PNIPAm, which stands for poly N isopropyl acrylamide. So this poly N isopropyl acrylamide is an example of a smart polymer. So why is it called a smart polymer? We'll be discussing it in some time. So coming to look at the structure of this PNIPAm, it looks like this. So it has an isopropyl group with CH3CH3, an acrylamide group, and a hydrophobic chain. So this is the isopropyl group, this is the long hydrophobic chain, and this is the acrylamide group. So this is the structure of PNIPAm. So this PNIPAm shows the property of LCST, which is lower critical solution temperature. This is why PNIPAm can be tuned to be a smart polymer. So what is LCST? LCST, or lower critical solution temperature, in context to this polymer is that at room temperature of 25 degrees Celsius or room temperature, PNIPAm exists as a solid state or as a liquid state. Now, when the temperature is increased at a temperature of 32 degrees Celsius, which is its lower critical solution temperature or LCST, this PNIPAm undergoes a salt to gel transition. At 37 degrees C LCST. So, this is why PNIPAm is actually a smart polymer. So, when the temperature is increased from 25 to 32 degrees Celsius, it undergoes a salt to gel transition. So, gel, it has a thick viscous jelly-like consistency. So, it is not in its solution stage, rather it is in its gel stage. So, temperature from 25 to 32 degrees and above, it exists as a gel stage. So this salt to gel transition is the property because of which this PNIPAm is reported to show this salt to gel transition, or that is why it is used as a smart polymer. So what is the physiology behind this salt to gel transition? So if we look at the structure of PNIPAm, we can see these are the hydrophilic groups. So if we are trying to solubilize this polymer of PNIPAm in water in its solid state, then this PNIPAm molecule will show hydrogen bonding with the water molecules. So once this molecule shows hydrogen bonds or forms hydrogen bonds with the adjacent water molecules, then these polymeric chains will be stretched in all directions. And once these polymeric chains are stretched in all directions and these are stabilized by the hydrogen bonds, then these polymeric chains will occupy a larger area, and therefore because of these hydrogen bondings, this polymeric system at a temperature at about room temperature 25 degrees Celsius, it will be existing as a liquid state or a liquid state. So once this temperature is increased from 25 to 32 degrees Celsius, we are providing energy in the form of heat. So once energy is provided to the system in form of heat, what happens is that these hydrogen bonds start breaking up because of thermodynamic reactions. So because of these different thermodynamic considerations of these parameters, these hydrogen bonds are shown to break at these temperatures. So with increasing temperature, these hydrogen bonds will start breaking, and as a result of which, because of this long hydrophobic chain, the hydrophobic interactions will predominate. So at 25 degrees Celsius, the hydrogen bonds will predominate, which gives the PNIPAm its a sol-like consistency at 25 degrees Celsius. But at higher temperatures, these hydrophobic interactions will predominate, and as a result of which these polymeric chains will start shriveling and these groups will start coming towards each other. So once this happens, the chain starts shriveling and it undergoes this salt to gel transition. So it will at 32 degrees Celsius, PNIPAm will exist as a gel. So now this property makes PNIPAm a very interesting polymer to work with. This is because this LCST can be, because it is related to hydrogen bonds and hydrophilicity and hydrophobicity at higher temperatures, this property can be tuned or modified according to our use. So tuning of LCST. So let me erase it so we can get some space. So there is this property called as tuning of LCST in LCST polymers such as PNIPAm. How this can be done is that upon introduction of hydrophilic moieties in PMIPAm, or by complexing the polymer with such materials which can also be carbon nanoparticles, these PNIPAm will increase the amount of hydrogen bonds in its structure. So, because of more number of hydrogen bonds in the structure, more amount of energy will be required to break them. That is why this LCST shifts from 32 degrees Celsius to 37 degrees Celsius, which can be tuned by the addition of specific molecules or specific elements in its construction, in its fabrication step. So this 37 degrees Celsius is the physiological temperature or the biological temperature, the temperature which is maintained inside biological systems. So at 37 degrees Celsius, if we can tune this LCST, if we can increase this LCST behavior, we can use PNIPAm for a wide range of biological applications. So we'll be looking at two potential applications or potential uses of this PNIPAm polymer. So one would be its application in engineering tissue culture approaches. So in engineering tissue culture applications. So if we have a basic knowledge about tissue culture or cell culture, we seen that when cells reach a particular confluency, cells need to be transferred or split or passage from one plate to multiple number of plates, so that after reaching this confluency, these cells need to be removed from the particular growth environment or the place in which the cell is grown, such as a T25 flask or a petri dish where the cells are grown. Then these cells are actually centrifuged, then pelleted. This pellet is resuspended and again resplit or passaged. So this process is known as passaging or subculturing. Of cells. So in subculturing, an important step is trypsinization. That is, the cells are treated with trypsin, which is an enzyme, to break the interaction of the cells with the substrate or the substratum. So most of the cells which are adherent cells, grow as monolayers. So adherent cells grow as monolayer in the tissue culture flask or the plate in which the cells are growing. So once that area is covered, then it stops or prevents the further growth of cells, because in such a flat surface, these cells will, or a two-dimensional surface, these cells will only grow on the surface of the growth plate, or the plate in which the cell is grown, and that growth will be in the form of a monolayer, or a single layer of cells will be growing on the surface. So in order to provide more space to the cells, these cells need to be removed from the plate and resuspended or reintroduced into fresh media and in fresh plates to promote or to enable their proliferation to take place. So, since these cells are monolayers, these cells have some attraction with the or attachment with the surface of the plate in which they are grown. So, these cell adhesion molecules or the molecules which enable the attraction to take place between the cell and the substrate, these need to be broken. And in order to break these, we use the enzyme trypsin. So, trypsin actually breaks these molecules which causes the attraction or causes the attachment of the cell to the substratum. Often it is found that in some cases, to reduce the use of trypsin, we can use smart polymers, or to eliminate this trypsinization step in cell culture, we will use these smart polymers. Now, what is the role of smart polymers in such an application? So, if we use PNIPAm in such conditions, so PNIPAm exists as a solid state at temperatures of 25 degrees or at room temperature. So, if we coat the surface of a plate with PNIPAm, then we are seeding the cells on it, then the cells will be incubated at a physiological temperature of 37 degrees Celsius. So at 37 degrees Celsius, this PNIPAm will exist as a gel state. So the cells will attach on the surface of the PNIPAm. Now, if we want to reculture, if we want to passage the cells or subculture the cells, we will simply take the plate out and incubate it at room temperature. So at room temperature, this gel to salt transition will take place and the cells will become in a suspension. So once the cells are in a suspension, we can easily centrifuge the cells. We can centrifuge the cells and we can obtain the pellet, discard the supernatant and again resuspend the pellet in fresh media. So this step might possibly be enough to eliminate this trypsinization process, which can be harmful for certain types of cells, because this is an enzyme which can actually break certain protein molecules as well, which might be important for the proliferation of the cells. So this might be a possible application in cell culture where smart polymers may be used to eliminate certain steps of this cell culture. Now, there can be another application, interesting application of smart polymers, which can be bacterial biosensors. So biological sensor or biosensor is a device which is used to detect any biological analyte. So bacterial biosensors can be fabricated using smart polymers such as PNIPAm. So what happens is that if we have a surface on which PNIPAm, a layer of PNIPAm is added or is introduced, so in room temperature it will exist as a solid state. So if that is inoculated with some bacterial species, then what will happen is that the bacteria will become metabolically active at 37 degrees Celsius or at higher temperatures, where the bacteria will start forming colonies, and then this layer on which the bacteria are growing on, or where they are introduced or inoculated, the temperature of that surface will increase. So here, if a bacterial biofilm is produced, the temperature will increase to 37 degrees Celsius, and PNIPAm will undergo this salt to gel transition. And this salt to gel transition, if we are seeing it, we can be sure that there is certain bacterial contamination in a particular sample, which is enabling the transition of this salt to gel. So it is a very fast process, because these bacteria can grow or can multiply very fast, right? So we do not need to go for any visual or spectroscopic examination for detecting these bacterial, the presence of certain bacterial species. We can just use a PNIPAm chip or a PNIPAm layer or a construct or a device which can directly detect this bacterial contamination by a simple phase change reaction. So These smart polymers have immense applications in different fields of biomedical engineering, diagnosis, as detection, as biosensors, and in several areas of tissue engineering as well. So, there are many other types of smart polymers, and a sound idea of these can be very helpful for designing a particular construct. So with this, we'll end today's video. Hope you enjoyed it. See you in the next video. Thank you.